Good morning, everyone. I, Chandraiah Coordinator and Assistant Professor, School of Pharmacy, PP Savani University. Welcome again, Day 3, Faculty Development
Your
Program
voice
on.
is bit down. Your voice is bit down.
Okay, okay. I think it's right now. Okay.
Clear now, yeah.
Yes. Thank you, sir, for confirming. So I welcome again Day 3 National Level Online Faculty Development Program on optimizing pharmaceutical innovation, ethical uses of AI for research writing, publication, and patent. So last 2 days we have gone through the power of patent through AI as well as the publication and writing, research writing. So today we are going to learn a new thing from our today's speaker, esteemed speaker, Dr. Chandraiah Godugu. Dr. Chandraiah Godugu is a distinguished assistant professor in the Department of Biological Science at NIPER, Hyderabad, renowned for his groundbreaking contribution to pharmacology, toxicology, and nanotechnology. With over 13 years of experience in research and teaching, he has consistently advanced the frontiers of science, earning him recognition among Stanford World Top 2% scientists for 5 consecutive years, 2020 to 2024. So Dr. Godugu academic journey begins with a Bachelor of Pharmacy from Kakatiya University, followed by an MS and PhD in Pharmacology and Toxicology from NIPER Mohali, where he focused on nanoparticle toxicology and advanced drug delivery system. He further earned his expertise during a prestigious postdoctoral fellowship at Florida A&M University, USA, where he worked on cancer drug delivery, 3D tumor models, and nanomedicine. Since joining NIPER Hyderabad in 2014, Dr. Godugu has led transformative research projects focusing on organ fibrosis, respiratory disease, and inhalation drug delivery system. His multidisciplinary approach integrates pharmacology, toxicology, and artificial intelligence, making significant strides in therapeutic innovation and toxicological prediction. A profile a prolific researcher, Dr. Godugu has authored over 200 publications, cited more than 7,700 times, with an impressive H index 45, reflecting the global impact of his work. He has also contributed to academic resources such as Remington's Science and Practice of Pharmacy and co-edited a book of nanotoxicity. His innovative spirit has led one granted US patent and three Indian patents alongside numerous awards including Dr. B. Mukherjee Award for Indigenous Drugs Research and DP Patil National Based Thesis Award. As a diplomatic of the American Board of Toxicology and a fellow of the Telangana Academy of Science, Dr. Godugu exemplifies excellence in regulatory toxicology and interdisciplinary research. His ability to secure extramural funding exceed 6 crore. Yes, more than 6 crore. He has get the grant and enabled the execution of cutting edge project fostering collaboration with prestigious institutions like IGIB, IIT in STEM beyond research. So Dr. Godogo is a dedicated mentor, having guided 60 master dissertation students and 21 PhD scholars, many of whom now hold position in the top global institutions such as Harvard Medical School and John Hopkins University. So Dr. Godogo leadership extend beyond academia, serving as a GLP inspector for DST and GCMA, a reviewer of over 40 international journals, and invited speaker at global conferences. So his contribution to nanomedicine, regulatory toxicology, and AI application is drug discovery have influenced policies and best practice worldwide. So Dr. Godugu vision for the future includes exploring phytopharmaceutical therapeutics, advanced AI-based toxicology models, and innovating drug delivery system, emphasizing translational research to bridge gaps between academia and industry. So his remarkable careers is a testament of his unwavering commitment to scientific excellence and societal impact, making him a relevant figure in pharmaceutical science. So with this introduction, I like to invite our today's speaker, Dr. Chandraiah Godugu, talk on the topic opportunities and challenges in drug discovery and development in the era of AI. Over to you, sir.
Good morning, all. Very nice to see, more than 144 people, joining online. I was told that, more than 300 people have registered for this particular, conference. Of course, not the conference, national level faculty, online faculty development program, organized by PP Savani University, right? Let me thank Samareswar Paul Roy for inviting me, and it was a short notice, and he just made a call, and he wanted me to be a speaker, and I happily accepted this. And also, principal sir called me. Bhavesh actually called me. That Bhavesh called me and requested me, can you actually become a speaker for this? And I was not sure what was the topic, but when they mentioned artificial intelligence and machine learning, so I thought, I can, because I have been talking about, and I was trying to, emphasize more on how, the artificial intelligence and machine learning is going to dictate the pharmacy, the growth. So, and very nice to see, your nice campus, your background. We can see, peaceful campus looks like, and yeah, it's a very great. Good morning again to all, and uh, thanks to all the faculty we have organized this program, as well as thanks to all the faculty who have been actually listening to my talk, right? uh, I'll just share my slides, then I think you can go ahead. And before actually going to my slide, um, I have I'm talking about my research in drug discovery and development, how AML can be used there, and some interesting data what we have got so far. We'll be discussing about it, and if you have any questions related to any of my slide, actually we can discuss, at the end of the talk. And if you want to disturb me, you just, is that fine if they want to stop in between, or they can ask
Can
later?
answer that. They can ask also, but not
Okay, so let
frequently.
let me just share my is whatever you think is good. We go ahead with that. Um, this is the one I'm going to share it, right? Um, yeah, sometimes we tried to put the full screen. I don't think so. It will come. It will be like this only. Is this
Okay,
fine?
it's fine, sir. It's fine. Completely fine. We can
Is
see
it
it here.
moving?
बीच में कौन सी
Uh,
प्रैक्टिकल
Visible, okay, sir.
Is it
visible is
moving?
not. No,
खाली
sir,
हुई
not
है ना?
moving.
Slides are not moving.
इसमें प्रैक्टिकल हुई है, दो या एक ही हुई है? 12 के बाद में
What did you say?
देखना एक बार कितने तारीख को हुई है?
Slide is not moving, sir.
Slide is
Not moving. It is fixed.
okay. Okay, that's it's not moving, right? I'm actually going to next slide. It's not moving, right? Okay.
No, sir, not
आप लोगों की
moving.
13 में थी,
Is it
10
moving now?
की 12 में थी, उसके बाद में
Is
No,
it
sir,
moving
not
now?
yet. No,
कोई
sir, no,
प्रैक्टिकल
sir.
इशू नहीं हुआ ना?
Okay, let me share again. Instead, all the technical issues will come. Let me
Yes,
share
yes,
again.
yes.
नहीं कर लो, अपना वो हुआ था क्या? प्रैक्टिकल तो हुआ था नहीं?
Professor, can you
Yes, yes.
बीच
mute
में
the
1
Yes,
तारीख
yes.
को
I'm just
other
trying. I'm
participants?
just trying.
एक मिनट
Just 1
Participants
second.
are requested to mute your mic,
Let me
please.
share again. Let me share again. Okay.
Dr.
All
Satyendra
participants.
Kumar. Yes. All are muted now.
We just tried, right? I'm doing entire screen sharing. Entire screen. I'm trying to. Or window. Which one I should?
You can try, sir. You can try enter screen also if you are comfortable. And
I think it's not allowing the entire screen.
okay.
I know it is there, but now is it sharing and moving?
No, sir.
Not moving.
Okay, sir.
It is
I
visible
can.
but not moving.
So I can do one thing, sir. I will make you the host. So I think on that case you can try. Is it okay, sir?
Yeah. 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 Please.
Okay, okay.
At the end of the talk, you can actually change the host.
Yes, yes, yes,
Yeah.
yes.
Sorry. I don't want to waste,
No issue, sir.
all
It's happened.
The
It is
slides are
faculty.
moving now, sir.
Slides is moving now. Yes, yes.
Sir, Your slides are moving.
Are they moving?
Yes,
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 sir.
yes, yes, yes,
Moving.
yes,
But am I sharing now?
Yes, yes.
yes. It's sharing.
Slides are
It's sharing.
visible.
Okay. And okay. I don't know what happened. Okay. Because of the host, he changed. Are they moving now?
Uh, no, sir.
No. When I'm putting a full screen, it's not moving. That's what the reason now. Okay. Now it's moving,
Now,
right?
now
Yes.
it's yes. Now it's moving. It's
I think we
perfectly
can
fine, sir.
go. I think we can
Yes,
only go ahead with this. Looks like
yes, yes. It's perfectly fine, sir.
second slide, third slide. No.
Right now it is the first slide only.
Okay. And now I think it's the full screen is not going. So kindly excuse me for this. Okay. I hope this will not discourage people.
It's fine,
Because
sir. We can clearly visible. The text
okay,
are clearly
okay,
visible.
okay, okay. So yeah, without wasting much time, let me jump into my topic. So the title, itself is very broad, right? Opportunities and challenges in drug discovery and development in the era of AML. So it's very broad. If you just take a discovery, it's a very big, area where there's a lot of biology, there is going to be a lot of chemistry, there is going to be a lot of pharmacology studies, and there is going to be a lot of safety studies. You'll be doing it in right development. When you talk about development, it can be detailed toxicology studies to prove the safety of the drug, as well as clinical development. So it's a very broad, right? And I strongly believe that AML can be used everywhere. How it is, because no, right now, at least in Indian pharma industries, whoever the multinational pharma companies like Novartis, Pfizer, what are the pharma companies we have, and Bristol Myers Squibb, BMS, these all what they're doing is they're trying to handle the bulk data in the pharma clinical trials. Basically, they're trying to focus more on clinical trials, how patient data, the clinical participant patient data, which is collected through different pages, can be handled through artificial intelligence and machine learning. Because humanly that is not possible, because each patient will be having a lot of data. And today, it's the data which is going to data is the most strongest innovation in the current trend. Because if you have good data, we'll be able to take the right decision whether to go ahead, or you want. So data is very crucial, basically, right? Now, whether it can be for the discovery, or it can be also for the development. So right now, all of multinational pharma companies, we are trying to implement the AML tools as a data science by using AML tools. That's one side of it. But we, the teachers, we the pharmacy faculty, will not be too much involved in the clinical trials, or such. It's industry activity, right? It's more of industrial activity. Yes, we can train the students so that they'll get a better job opportunities. But what as a researcher, since you try to teach B pharmacy students, M pharmacy students, as well as PhD students, um, whereas the ways the AML can be used in the drug discovery, I feel drug discovery is the area we can actually put more efforts from the faculty side, at the academic side, right? So just going back to the difficulties we have with drug discovery and development. And if you see at a glance, it's very time consuming, right? 15 years, its cost is 2 billion. Or earlier, 10 15 years back, we used to say that it's a 1 billion. Now it's a 2 billion. Now even some of the reports say that it can also go to 5 to 6 billions also. Money is involved, as well as the 10 years. And time is money, so you are losing the time as well as the money also, both. So why Indian pharma companies are not doing a discovery? Because of this factor. We think that no, we are not certain, because it's highly risky affair, and we are not sure whether we'll be able to be successful or not. Why you want to take a risk? And if you are failing in the eighth year, or if you are failing the 12th year of that discovery and development process, and it's a whole company fate will be reversed. And we have seen certain disasters happen to some of the organizations. If you see Lupin, oh, no, nothing happened to Lupin, but they slowly slowed down the discovery phase. And the Day-3 used to do a lot of discovery, and they had a setback of one of the compound, which was an anti-diabetic compound, was having a safety issues, was causing a carcinogenicity in one of the long-term toxic studies. They had to retire the molecule. Then actually they moved away from the discovery for some time. Later they started a subsidiary company called the Origin Discovery. Now it's the Origin Oncology. It's doing wonders now. And now actually the Origin Oncology, which is also Origin Discovery, which is a sister group of company of Dr. Reddy's. If you get a time, just go back and see its programs. It has more than 15 molecules actually advancing to different phases of the clinical trial. And I had a opportunity to interact with them, and they actually very rigorously implementing using the AML tools or modeling tools to design the molecules. Basically, the source of the drug we all know. I don't want to bore it. Now, earlier it was the plants or the biological origin. It can be a synthetic origin. We all have all the three organs. Now it's the era of the biologics. But now we can also generate the drug from the computational ways. How do you generate the drug from the computation tool? That's where artificial intelligence and machine learning can be part of it. In the sense, if the drug is already approved, you can actually analyze the whatever the reported data, say that no, yes, this particular drug not only inhibiting, not only causing this particular pharmacological benefit, it is also maybe useful, because when I analyze the data collected from different preclinical as well as the clinical studies, we have observed that there is certain phenomena happening it. And if we did the farm proteomics and genomics, we understood that this particular drug, which we have not noticed earlier, is also causing another benefit, that is actually repurposing. So for the repurposing also, you can use the artificial intelligence and machine learning, because again, there also, if you take the proteomics or genomics data, it's thousands of data sets from each group. You'll be having thousands of parameters on how they're connected, how this networks is established, informatics, bioinformatics. So because though we have bioinformatics tools, if we can also apply the artificial intelligence, because artificial intelligence is talking about, doesn't matter how much is the bulk is the data is, if we can able to put the meaningful input into the computer or into the machine, it is going to make a meaningful data sets. In the sense, okay, when I gave this drug, this set of proteins are altered, this set of genes are altered, this particular safety issues have been encountered. Based on that, we will be able to understand by using a machine tools like artificial intelligence and machine tools, that you will be able to better get a clarity compared to humanly, because when you have to handle humanly, thousands of data sets cannot be handled, and you have to spend a lot of time, and human error is going to be huge, right? So you can even use for the repurposing as well as even for the new drug discovery also. So when you talk about the new drug discovery, and you will be starting with the target validation, and I feel target validation also can be using the AML tools. How again? Because every animal study, every cell culture based study, every clinical study reported in the literature, they are going to deposit the data sets in the data banks which are available online. Now, for you to select a target, how do you pick it? Will you go through all the lakhs of data sets? Not possible. That's where you can actually train your computer to say that pick this particular the patterns, so that you come to know in a cancer, these are the certain kinases, or these are certain receptors, actually how they are actually changed, how the expression is changed, and upon the disease as well as the normal. So you will able to get the meaningful data sets in quick time, like you are using ChatGPT. That's also right. Without reading the articles, we'll able to get a synopsis of it, just to have an idea. That's this is for the short-term use, but for if you're talking the drug discovery, this is not just for reading purpose, right? Actually, you're trying to make a big call based on the molecules, targets suggested by your AML tools. So target validation, where you can think that we can take a call whether your target is proper or not by using AML tools. Lead optimization, hit identification as well as the lead optimization. If you see 10,000 molecules, 20,000 molecules are required for you to get a proper lead. Now, how do you do this? Lead optimization. Now we know all the molecular modeling tools. We also know all the docking tools. We also have a lot of computational tools. These all actually based on the AML only. In the sense, whatever the data we have so far generated from different chemicals for different proteins, one side, and also we also have a protein crystal structures through your crystallography and other techniques. We have protein structure with all the amino acid sequence and all the arrangements. Can you able to arrange a complex protein humanly without emission? What kind of binding pockets it has? What is the active site? What is the binding site? What is the inactive site? And what is the catalytic site? Will not. So even for you to get a proper protein which is required for you to do the docking, you will be using your artificial and machine learning. That means everywhere you are going to apply it. That means you can use for the protein structure finalization AML tools, even for the lead optimization. Because if you are not properly designed your target, that means your G protein couple receptor, which you think is your new target to be used for the discovery of the new molecule, and if the protein structure is not properly designed or not properly framed, or what you call arranged to study the to do the docking studies, you're actually doing though the protein is of important, but the structure you are using in the computer to design the molecular modeling will be wrong. Structure will be getting it. Understand what I'm saying? What I'm saying is, even for the protein, the proper conformational arrangements with all the active sites and the inactive sites to be which are the which is really having a role in particular disease condition needs to be properly captured, include even the ion channels also. So one side is the target, which is mostly the protein. Other side, which is the leads in the sense, as I mentioned, 10,000 molecules to 20,000 molecules are required. Now, earlier, when you don't have the these tools, we have to synthesize 10,000 molecules. Just imagine how much time is required. 10,000 molecules, chemistry, how much efforts they have to keep it. And each chemistry will be different. So you need to have different starting materials, and you need to synthesize them and characterize them, and so on, right? It was taking a lot of effort. And ultimately, since we have no idea whether, because we never studied the interaction between the protein and the your NCE, you will be always getting a lot of inconsistency in the results. Now, what is the advance now? We have a molecular modeling and a computational tools where, without synthesizing the molecule, just taking 100 to 200 molecules, you will be able to study how they are interacting with the protein. Your molecules you have designed, how they're interacting it. And this 200 molecules, you are not just randomly picked it. You have picked up based on the huge data sets which are talking about, okay, this amino acid, Lipinski rules you are going to apply, all the ADME profile you are going to apply, all the toxicology features you are going to apply, all the physical chemical properties which are required you are going to apply, so that you know that, no, okay, I need this such kind of features. Can you do it again humanly? No, you cannot. That's why you are again using artificial intelligence, machine learning. What is the basis for you to take it? Because that data bank which is already available, the literature which is already available, very well integrated into your this molecular modeling tools like Schrodinger, I know, Autodoc Wien and other software, right? So that means you are going to, before even starting your pharmacology, before even doing the chemistry, you know that you have a very interesting molecules. This is how now we have to think of the discovery. Earlier, it was just selecting some molecules, synthesizing it, later doing a pharmacology and doing something else. But now you need to at least predict it. 50 to 60% prediction can be right now. Within a couple of months to maybe within less than 1 year, we will be able to optimize the chemical structures which are going to be probably leads. Now you will be trying to do your biology, let's say. That's what you will be doing the bioassay and so on, right? Then once you have the lead molecules optimize. You'll be going for preclinical testing, both in cell culture as well as the animal models for pharmacology. They're followed by detailed safety studies. So generally, you'll be taking 2 to 5 molecules. From 250 molecules, 2 to 5 molecules you'll be taking for the preclinical animal pharmacology and toxicology studies. And you all know the clinical trials will be done first in human dose and so on. And ultimately, once clinical trials are giving a positive results, you'll be saying that no, this can become a drug. You'll be again going for the NDA. So drug discovery taking 2 to 6 years, and the development is taking 5 to 10 years as a range. Again, it depends, what kind of category of the drug you're talking about. If it's an anti-cancer drug, you may get a quicker, approval, because you can actually bypass some of the toxic studies, because assume that risk versus benefit, where cancer is more riskier compared to the safety we are talking about of the drug which we are proposing it. So it can be it may anti-cancer drugs, the developmental phase will be relatively faster compared to the anti-cancer drugs, or mainly if it's a lifestyle related disease like diabetes, hypertension. You will be needing more the detailed toxicity evaluation, because you are going to expose to the drug for longer time, and that's where you have to do more safety studies in the animals as well as large animal models. Now, same thing I'm putting in here, a target selection. You can actually the cellular and the uh, other proteins, and you can use the genomics, proteomics tools to select the target. Then followed the lead discovery. You will be using a synthetic ways, combinational chemistry, and assay development, and all you will be doing it in this stage. Then followed the medicinal chemistry, because medicinal chemistry role here is to now do the structural activity relationship. What kind of SAR you are getting it and all, so that now you will be also trying to optimize the ADME. Then you will be doing in vitro studies, in vitro at the enzyme-based assays or kit-based assay, followed by the cell culture-based assay. You'll be doing it to understand how the medicinal chemistry we have the chemistry we have developed, the lead molecule is behaving in the in vitro studies. Then followed by in vivo studies in the animal models for both pharmacology, toxicology, and finally clinical trials. So if you see, the drug can come from the biological products, as well as the drug can also come from the synthesis. Now the trend is of the biological products, but as of now, it's the only industries who are actually actively working on it, because biological biologics, the expertise needs a sophisticated laboratory and so on. So let's focus more on the chemical synthesis, or our small molecules. So when you have these molecules, you'll be trying to do the pharmacology toxicology screening. Once you have optimized the molecule through computational tools and AML tools. Now then you'll have the pharmacology toxicology screening, and then the next thing is the compound biologically active or not? Either is the compound is safe or not? Both are important. The compound is not biologically active, there is no point taking it further. You will have results. Now, similarly, if the compound is biologically safe or not, the compound is not safe, you cannot advance the molecule to the next level. Now, if the compound is safe, what is the dose response curve for the pharmacology and pharmacokinetics and pharmacodynamics? For activity and safety, you have to establish. The results will be discussed in the team meeting. Very important, because the team meetings are very, very important drug discovery and development, because that is the one going to this is a multidisciplinary team again. All the chemists and pharmacologists, formulation scientists, all will come together. Then whether to go ahead with the what are the pharmacology toxicology data you have or not, you will be taking a call. If it is to go, it will become a lead compound. From the lead compound, you will be doing the preclinical testing, then followed by clinical trials and so on. If it is no go, that's where you are going to kill the compound. No go is nothing but killing the compound. So that means early killing is the best killing. If you want to kill the molecules, kill early. Don't advance it to complete toxicology profile and say that no, it's a toxic. If we can use the computational tools, if we can use the AML tools, and we can able to say that no, yes, this molecule could probably be a toxic, because I have used all the computational tools. I have also designed this molecule. I have also used my AML techniques to understand whether my chemical structure, which is I'm trying to make it a new drug, is going to be safer or not. You can able to take a call. You can actually kill the compound early. Early killing is the best killing. Okay, if we are not killing it, if the molecule is moving from preclinical to clinical, then followed by FDA review for the NDA, that can be getting approved. So this is one side, right? Now, if you see the costs are very high, drug discovery development costs are very high. Why costs are high? Because you need to do more toxic studies, as well as the preclinical costs are high, as well as the clinical costs are also high. So preclinical costs are high. Why? Because increased battery of the toxic studies. Before thalidomide, number of toxic studies in 1960s, number of toxic studies required were very limited. We don't have range of toxic studies to be done. Now, if you take the ICH safety guideline, S1 to S11 guidelines, you have range of toxic studies to be done, safety pharmacology, reproductive toxicology, and so on, local toxicity, sensitization, and so on. So, range of toxic studies are increased today compared to 50 years back or 10 years back. Your guidelines are only increasing. The moment you are getting more guidelines means you have to meet the guideline requirement and perform the toxic studies as per that. And now, earlier there was no concept of the GLP. Now we have a concept of GLP. That means you have to do the toxic studies, preclinical studies in the GLP labs. The moment you go to GLP labs, the cost of the same non-GLP study to GLP can go to 5 to 10 times higher, because GLP studies are very quality systems. So more documentation, more protocol reviews, more data. You have to store the data for 9 years. That's compulsory. That's why because of all this requirement, the preclinical toxic studies and preclinical pharmacology studies in GLP labs can increase the cost from five times to 10 times. But now it's a requirement. Earlier it was not a requirement. Of course, if even the regulators are not asking this particular GLP toxic studies, but you will still do it because you need to have a confidence. You don't want to take any risk of your molecule showing a toxicity later in the clinical trials. If the molecule is showing safety concerns later in the clinical trials, it's a big setback to any organization, because you might have invested a lot of money. You might have already spent 6 years of time to come to the stage, and also you already initiated the clinical trial. You know how the clinical trials are expensive, right? Evidences and factors affecting growth in the clinical trials. So now, if you compare to earlier, you need to do the more clinical trial subjects, increase complexity and more procedures required for the patient. Patient recruitment and retention procedures are now very road. It's not that you have taken the clinical trial participants and given the compound and 1 week is over. You need to follow up the compound the patient for lengthier period of time, saying that okay, because of that particular dose he has taken as a clinical trial participant, what kind of the lifestyle is going to have, what kind of safety or issues he may be encountering it. You need to monitor them properly. And also, if it is a long-term chronic disease condition, you need to have more follow-up needs to be required. And now, if you have to do the clinical trial, you need to compare the comparator drugs. So the moment you add the comparator drug, another almost 20 to 30% of the cost is going to be increased in the clinical trial, because you are forced to compare with the existing drug, with your new drug. So cost is only going to increase. So these are some of the setbacks, right? Now, if you're having so many hurdles, if you are having so many limitations, that's why all our Indian pharma companies, we call it all the major pharma companies except the Origin Oncology and some other companies, have already safely, conveniently moved to safe game, which is making a generics or making a formulations, just only supplying the drugs to the developed countries. So, and we don't want to really discover the drug. This is the biggest challenge now, because until unless you don't get the new drugs, the drug which is approved in the US may take a lot of time to come to India. Even if it is coming to India within 1 year, in case if because of need, because of they have innovation, or they have the rights on the drug, they will be dictating how much is the charge they'll be charging on the drug. The cost will be dictated by them, right? So like iPhone we are using today, just imagine the iPhone we are procuring today, we are paying money to yes. How? Because they are the innovators. Similarly, if the new drugs are not discovered in India, for at least next 20 years, those drugs, whatever been consumed here, we are paying the taxes, or we are actually giving the money to the company which is discovered innovator. So why Novartis, why Bristol Myers Squibb, why other companies are trying to have a local office here, and they're not doing a discovery here? Remember, they're not doing discovery here. They're only using our excellent pharma setup as a manpower for them. It's okay, we are getting an employment for our pharmacy candidates, but is this sufficient for us? Do you think? No, it's not sufficient. So many pharmacy colleges, more than 1,000, if I guess. What are we doing when we are not thinking of, when we are not stimulating the people to go for a discovery and development? We only wanted to work in an organization where I sit on a computer and try to only do the data analysis, and I don't want to really design the new molecule. It's a crime. Actually, it's actually lost to our country, basically, because it's expensive. Pharma organizations, they cannot, pharma companies cannot do it. But we people sitting in the academia, we have chemistry, we have a pharmacology, we have a formulation, we have everything, right? Why can't we at least think of using the current tools like AML, so that no initial studies, you are not daring to go for clinical trial, no need to do the clinical trial. Just have a proper chemistry design, use the tools which can actually give you a proper chemistry design. First understand which is the best target for you. Again, use the tools, what are the tools you can use it. Once your the target is identified, the target can be a unique and which nobody else is exploring it. Then followed by try to get its protein binding domain structure, PDB structures. Then followed by you try to dock the with the existing the molecules, and try to improve. Now once you get a hit molecule, now try to optimize the hit molecule. So once we have hit molecule, it is means it's just binding to the target only, but it's not the optimal molecule, it's not the final molecule. From the hit you can make the lead. For you to make the lead also, you can use the competition tools. You can also the artificial intelligence tools. What are the different AML tools are there? So you give the inputs. I wanted this particular feature in my compound because this particular heterocyclic, this particular ring is required, this particular bond is required to bind to the protein, which is to inhibit or activate it. Now, to improve the solubility, to improve the protein binding or whatever it is, improve the off life, how can you further improve it? We can do that, right? So basically, that's where, as academic institutes, if we can think of, it's possible for us. It has to be interdisciplinary. You have to collaborate together. You need to have one common goal, which is one target, and multiple teams try to come there. So through that approach, I feel we can actually uh, design a molecule, synthesize a molecule, screen them the pharmacology model, and also do some of the safety studies and say that no, it's reproducible. Yes, what I have observed is it's reproducible. It can be if I repeat multiple times, I'm going to get the similar type of pattern. Now you can actually and you compare now that somebody else is working with this target, or what is the what is actually what is the status of this particular disease. Are there any drugs in the already available in the market? You have to take a disease where there is unmet medical need, and also the target you are taking should be unique, where you can you are the first one, or you are very few to start working on it. Now people will take your data to further develop it, because you cannot do any further GLP tox studies on your own until you get the funds. I will tell, at least you'll be needing 10 to 20 crores to complete the GLP tox studies. 10 to 20 crores. And I'm sure if we have the preliminary data like this, where you have carefully selected the target and have the up to the lead molecule synthesized and done some pharmacology, I don't think so you will have a difficulty getting a fund. Doesn't matter you are from a government institute or private institute. You should be able to convince that no, this my data is so and so. You take a videos to demonstrate that how a compound is protecting it in the animal model or whatever it is. I think we'll be able to convince the funding agencies to get the required funding. Once you do the very detailed toxic studies, now it's a proof of concept is established for the pharmacology as well as safety. Now it's up to you how you want to collaborate. You can go to now pharma companies to collaborate further. You can propose to them that so and so we have developed it. This is the data. Now because the funds are not, we cannot handle it, the clinical trials. Can you take this and further develop it? So it's always possible, right? Why we cannot think of, let's try. That's what is really required. It's not just a publication, not just taking one compound and doing it. I think we can do much, much, much better than what we have been doing today, because of the number of pharmacy institutes, number of faculty spending the full time in this profession. If you are not daring, I think we are really, not justifying to our time, because we have put a lot of our energy and time to get our doctorates. It's not joke. It's not easy, right? And why we should be just working for some salary, or why we should have, beyond that, right? So what I feel is time and cost can be reduced through the AML tools. Improved preclinical testing can be done. How you're going to do the improved preclinical testing is because you already optimize the molecule at the computer level, even without doing it. Efficient clinical trials very much possible, because no, the design can be better handled. Lead discovery, as I mentioned, is very much possible. So these are the some of the AML tools people are using it. Deep talks. What is deep talks? So people have developed certain tools to optimize some one aspect of the discovery. Now deep talks about what? It is going to talk about the toxicology. Now when we are trying to design a molecule, you can use such kind of the deep tox tools to say that no, because safety is at most important compared to the pharmacology. Because if you have any concern with safety, nobody will actually uh, allow to further progress it. Pharmacology, we assume that yes, it may have activity, because you have already taken care of it. Safety is the one going to create a lot of trouble while you are trying to take the molecule to the next level. So deep tox is one tool. No, it's a AML driven tool. I have not used this tool, but actually I got this particular information from some other publications. Delta Vena and potential net. Again, it talks about the molecular interactions and all the informatics tools. Deep chemistry, and it will give you chemistry, because no synthetic tools also. Chemistry, designing the molecule as well as synthetic modalities also can be for done. Like that, you have other tools as well, right? QSI tools and AlphaFold. Very important one is AlphaFold. Now, if you see the Nobel Prize this year has gone to one of the person who was contributing to the AlphaFold. Why this is? Because Google, the person working in the Google, we all know that AlphaFold is the protein structures in Google available, but we are not realizing it, right? Why Google is investing a lot of efforts to get protein structures in the Google platform? Because they realize that no, the protein structures are the ones very crucial for any pharmaceutical discoveries to happen. Now, if these carbon proteins are available in the internet, right? Freely, right? You can take the structures, play with them, dock with the existing molecules, try to design your own chemistry by using chemitron and whatever it is, and see how they're binding it. It's just you play with them, right? What is it? We don't lose anything. Now, again, as I mentioned, iPhone, same thing is happening here. If you see it, uh, the first, I think it's regional regional performance investments, and these are different measurements for AML. AML based activities are mostly dominated by the USA. That's what I'm trying to say. Followed by Europe, followed by UK, then followed by some other countries. Where are the India is Asia, and India is not even India. And one fifth of the population, one fifth of the population is India. And where are we? We have so many pharmacy institutes. We have excellent expertise in the computers and computer engineering and all. We have IITs and high quality institutes, and that's where there is a big opportunity for us. Now, a pharmacy person with a AML background, tech person, if they can sit together, define the problem, saying that no, I wanted to do so and so. Actually, the tech people, they don't know. They don't have a lot of knowledge on the pharmacy, what is required and what is. If you can able to define your problem, okay, I wanted to have so and so, and this is the my requirement. I want. If you give the roadmap, that's where you can do it, right? So India is relatively doing better, but again, discovery part is still missing it. So again, here US is dominating it. So why it's superpower is because they believe in the innovation, and we actually so far have not done justice to our potential. And compared with China, just see the China now. Why America and China is fighting is because China is much ahead of India and US in the technology and innovations are they are much ahead of any other country. Now, if you see the some of the examples are companies which have progressed to if you see there is a Excentia, second and third are the Excentia. This is the company, the most it is the first AI based company started, and if you see their molecules are already moved to phase 1 and phase 2 trial. Even you have in silico medicine, this also moved to phase 1. I think now it's in phase 2 trial. Like that, there are already companies working with the AML tools to discover the new drugs. It can be repurposing or it can be designing the molecules. So basically, from 10 years, probably you can reduce all the first 6 years of time. Clinical trials, you cannot reduce the time. We can reduce the time, but you cannot completely avoid it. You need to test in the clinical trial, but at least 5 to 6 years of time can be reduced, so that your failures will be known initially only, so that no, you will not be getting any surprise later that okay, this molecule is having a safety issues. You will not have that particular issue. So these are some of the examples. Now this figure may be very crowded. The left side I'm talking the Roche, Pfizer, and so on, right? All the Sanofi, Eli Lilly, these are the companies. Now what they're doing, they have a collaboration with the AML based company. Why Eli Lilly or why Pfizer or why Merck is trying to work with the small pharma players? And this is also, we are also recently also seeing a lot of collaborations happening with the multinational pharma companies. Very successful pharma companies are trying to go to small AML based organizations. We have expertise to give a support to the large pharma, multi-billionaire pharma companies, so that they will be able to better do the pharma innovations. When all the pharma companies are trying to collaborate with small AML based companies, why we can also have a small startup in India is also really exactly doing the same thing. So there is an opportunity. I think you can go through the details. What kind of collaboration they are doing it? It can be discovery, it can be development, but actually there is a lot of collaborations are taking place. They realize that without AML, they will not able to do the discovery in an efficient way. So this is the background. What I'm trying to convey here is it is going to have a very crucial role. It is going to have a vital role. That's why we all need to be prepared well. So what is required from our side? We need to have the basics of it. You need to define a problem, what exactly you want to do it. It's very fancy what AML tools. Everybody is using it in publications. Everywhere we are using it. But that's our purpose is different. It's not just a small cause. Our purpose is a bigger cause, which is a discovery development. We want to do the innovation. That's why we need to have a basics learn wherever what are the courses required, what are the. See, it's possible now. It's internet era. We can get any information. You can enroll for any course. Have a basic certificate. Learn ABCDs initially. Then there are certain program needs to be learned. Once you learn that, you are very good at pharmacy subject. If you can put together, also take some of the success stories of the how the people are doing the drug discovery, possible to have a better success. With this background, now I'm going to move to how we have used some of the computational tools, how we have used some of the pharmacological tools to the drug discovery and development. So you know that novel targets are required, and the natural products also can be one of the source for you to get the drugs as well as biologics. So I'm talking about the natural product nimbolide, which come from the neem tree, and we all know how important the neem is, and herbal products. We generally believe that much safer compared to the rest of the molecules. Neem leaves contains the chemical called the nimbolide, which is excellent phytochemical, and we have been using this chemical for and doing a lot of pharmacological studies. We have more than 15 publications on just with one compound, and we have proven that now this is where the computational the molecular doctor studies we have done it. We proved that nimbolide was actually inhibiting the TNF alpha. We know the TNF alpha what it was doing during COVID, because TNF alpha is inflammatory mediator. So we are struggling to inhibit the TNF alpha, and doing at the same time only in before COVID only we are doing the studies how the nimbolide is actually inhibiting the interaction with the TNF alpha. It was exactly inhibiting it, and we have done the animal studies as well as the cell culture based studies, saying that not TNF alpha expressions at TNF alpha was inhibited by the nimbolide in the animal model and the mice model as well as the cell culture based models. So we have done the different pharmacology studies and like in vitro studies, because we have taken the raw macrophages as a THP1 cells to understand how nimbolide is anti-inflammatory. The green fluorescence indicates the inflammatory response after LPS lipopolysaccharide stimulation, and nimbolide actually reduced in the green color. And the red color indicates that it's the reactive oxygen species are actually happening released from the mitochondria. The source of the reactive oxygen species are mitochondria in both raw cells as well as THP1 cells, and it was controlled. The levels, ROS levels are in mitochondria, and the total cell are controlled by the nimbolide. And we have also done the lot of protein expressions, because just doing a DCFD assay is not sufficient. You have to do the protein expression, saying that what protein is altered. If until unless you don't prove your mechanism, nobody will take your this thing. And how did you do the animal study? The animal study for the ARDS, acute respiratory distress syndrome or pulmonary fibrosis, we have very good models here. So in this particular animal model, what we do is we try to keep the inducing agent. And for ARDS is LPS and pulmonary fibrosis is bleomycin. We try to keep this chemical directly into the lung. How do you put into the lung? So that's why we have called the oropharyngeal administration. We can actually do this. We have paper published on that. So then we have confirmed whether lung is developing a disease or not. So if you see the BLM is nothing but bleomycin, it's clearly changing the lung histology. So very important to have a proper animal model, right? So this we have it. Not only that, we also have a model called the whole body plethysmography. This is nothing but how the lung function can be. So you keep the animals in these two glass chambers, and we'll try to study how the lung functions, the vital capacity, tidal volume, and inspiration, respiration volume. These all can be measured by just keeping the animals in this and stabilizing them. So then all the breathing pattern all can be studied. Why is required? Because we are working the lung. It's not that lung will give you every information. These are the functional parameters. You can able to get some information how lung is behaving. Very good information. So what you are saying, we have disease model. You have given a compound, what the nimbolide, whatever it is, and you can study how it is impacting. Similarly, we also done the histopathology studies with the nimbolide. If you see the LPS group, the HN staining and tolerant blue is the tolerant blue staining. So severe inflammation is seen in the LPS groups, and nimbolide pre-treatment as well as the concomitant treatment up to only 3 mg per kg is controlling the inflammation. So very good, right? Lung inflammation is reduced in the mice model because it is inhibiting TNF alpha. So here we are proving that one only. Nimbolide, by inhibiting the TNF alpha mediated NF-kappa B and HDAC nuclear translocation. That means NF-kappa B and HDACs are the transcription factors. Actually, they need to go to nuclei to induce more genes which are involved in the pathology, which is in the cytokine production. So nimbolide by inhibiting the TNF alpha is preventing the nuclear translocation of the NF-kappa B as well as the HDAC. That's how it's actually controlled. This is the mechanism we have proven it, and we have proposed this particular through our experiments. We have proposed this, that no, nimbolide is inhibiting TNF alpha and HDACs, so that it's prevented the inflammatory cytokines. So we have published this work. And if somebody wanted to read, this paper is freely available. If you just keep nimbolide and TNF alpha, you will get from the internet. It's very good journal actually. You can follow it. Since nimbolide was inhibiting the TNF alpha, we also done another study in the psoriasis. So it's imiquimod-induced psoriasis model. This is the normal HN staining, and if you see the imiquimod-induced psoriasis, you can see the epidermal thickness is increased. That's also called acanthosis, and we can also see the thickness is increased in the imiquimod disease control group. And treatment with the nimbolide has controlled it. This is the psoriasis score, which is higher in the disease control, and the treatment with the nimbolide has controlled it. So we also done the protein expressions to understand whether what we observed in the histology also done through the immunohistochemistry. This is immunohistochemistry data. However, we have a problem with the nimbolide. It's a poor water solubility. It is not orally bioavailable. It is also having a, and what are the activity we have seen are only because of the parental administration. That means you need to inject the nimbolide. But the problem is, can you give nimbolide, a natural product, intravenously? Nobody would like to take it, right? You never you never accept a natural product to be injected into your body. You only your mind is very well fixed that no, it's a natural product. I only want to take orally. So that's the problem. Now we have also done the pharmacokinetic evaluation in the nimbolide. This is the LCM by LCMS method where we have. Okay, let me tell you, nimbolide we have not isolated. We have procured from a person who is actually supplying to some pharma companies. So we have taken them, procured that molecule. Surprisingly, nobody have done the pharmacokinetic study with the nimbolide, which is very essential, right? What you do, you give the compound to the rat and collect the blood samples and estimate the quantity of the drug present in the blood at a different time points by the LCMS method. So this is how the LCMS is more of a mass by charge driven quantification. You have done it. If you see this pharmacokinetic profiling of it, the first figure is actually the nimbolide oral administration at 10 mg, 30 mg, 50 mg. The y-axis is only talking about the nanogram per ml concentration. Only maximum concentration is 35 nanogram per ml. Only 35 nanogram per ml is the highest concentration you can see as a Cmax you can see in the blood. It is sufficient. That means very limited. Whereas the IV group, you can see 5,500 nanogram per ml because IV directly into the blood. That's expected only. So then based on that, we can see that no nimbolide oral availability is less than 3%. So that means that's why you cannot use orally the nimbolide. Now what is the solution for you may have to make a formulations or you may have to make the analogs of it. So that's what we have done it, but I'm not going to cover that part. So this is one part where we have used the natural products. The one important the area we are working now is in the netosis. So it's a COVID. They also have a lot of inflammation in the different organs. We don't want to talk about it. But if you see in the COVID or in a lung failure, we have seen during COVID was inflammation, severe inflammation. Inflammation was the big villain in the COVID related deaths because it's not the virus which is causing a havoc, because virus is a foreign particle. It was triggering the immune reactions, inflammatory responses. Due to inflammatory responses, your organ is activated for the inflammation, and the protective inflammatory response is aggravated, is causing the inflammatory cytokine storm. So this is actually mediated by a process called the netosis. So netosis is nothing but neutropil extracellular traps, which are the a fragment of the DNA which is separated from the DNA, is coming out along with other biochemicals to form a structures called the nets. So the process is called the netosis. So what we are saying is netosis is the one of the important event, because wherever there is inflammation, the neutrophils will be going to the side. When they go to the side, they try to release the nets. So these nets are acting like inflammatory mediators. What we are saying that there is a way you can prevent the netosis process, so that we will be able to control the inflammation. So again, this slide is talking about how the different COVID complications, short-term complications, immediate complications, as well as long-term complications are linked with the netosis, right? So now, if you see this cellular events, the nets which are formed in the neutrophils are mediated by the enzyme called the PAD4. So what you should know is PAD4 is the enzyme which is required for the netosis to happen. That means, can this PAD4 enzyme is an ideal target for you to inhibit so that the netosis process is not happening, not taking place? PAD4 is peptidyl arginine deaminase 4 type 4. That's called the PAD4 enzyme. So what we did was we tried to, so now the netosis is having a role in different sepsis conditions, arthritis, and fibrosis, as well as ulcerative colitis during COVID. This is nothing to do with COVID, but in COVID we have seen a lot of inflammation. That's why I'm talking about COVID. Now, the PAD4s are the enzymes which are responsible to the formation of the netosis. Now, these PAD4s are getting activated by the reactive oxygen species. If you remember, we have also done the reactive oxygen DSA. So that means when there is a stimulation of the inflammatory cell accumulation in the site, they'll be release the reactive oxygen species. The reactive oxygen species will be trying to activate the PAD4. So this PAD4 enzyme will be trying to scintillation of the histones. Histones are the nuclear proteins. So due to the scintillation, the some part of the DNA will be decondensed. That means it's coming out from the nuclei and uh, released out of the cell, and that is called the netosis process. So that means you can actually have the PAD4 inhibitors to control the inflammation, as well as the once nets are formed. I mentioned nets are actually nothing but the extracellular DNAs, the DNAs, extracellular DNA, right? Now, if we can break the DNA, the DNase is the enzyme, and DNase is the enzyme which is going to break the DNA. So there is already a drug approved called the Dornase alpha, also called the palmozyme. This is a drug which is already FDA approved, which is used in the cystic fibrosis, because cystic fibrosis, the sputum is very thick. Why it's very thick is because there is a process called the netosis there. Because the netosis, because DNA samples, DNA is highly viscous in nature. Due to high DNA in the form of netosis in the sputum samples, they're very thick. Because the sputum is very thick, they are going to clog the respiratory bronchi. So that now, since nets are there, extracellular DNAs are there, if we can give the enzyme called the DNAs, also called the Dornase alpha, it's going to break the nets, so that no, the sputum will be loosened. This is what the background of the using this particular DNA. So now see the link. Netosis is the process which is involved in the neutrophils activation, followed by the DNA removal from decondition of the DNA from the one, and this is actually triggering the immune reactions one side, inflammatory reactions one side, and it is also having a role in the cystic fibrosis. So the drug is already approved. Now, can you use this Dornase alpha in the in your to control the netosis? Is the another investigation. So what we did was we made a one nano formulation which is trying to coat with the DNase or Dornase alpha, and we also try to use the PAD4 inhibitors through our discovery approaches, so that no, we can evaluate them. So we have also done the animal studies in like we have done the pulmonary fibrosis study, because the moment you have a acute severe inflammation in the lung, which is going to cause the fibrosis in the lung, and if you see the PAD4, which is an enzyme found to be increased or expressed in the pulmonary fibrosis compared to the normal cell. That means it's upregulated in the disease conditions. So this is what I'm talking about, pulmonary fibrosis, which is a normal lung, where you have a very thin junction between the alveolus and the capillary. But due to fibrosis, where your collagen accumulation is more, this will be highly correlated with the collagen extracellular matrix. Due to that, your oxygen and CO2 exchange will be not proper. That is what is called the idiopathic pulmonary fibrosis, or the pulmonary fibrosis impact. Right. So we already have a PAD4 inhibitor, which is experimental compound. It's a chloramidine. We have demonstrated that chloramidine is controlling the pulmonary fibrosis. This second column is HNA staining, mesenteric staining, picrocytic staining, and polarized microscopy histological images of the bleomycin-induced pulmonary fibrosis groups, where chloramidine treatment has reduced the this extracellular matrix accumulation. We also quantified those markers. What we are saying is a known PAD4 inhibitor experimental agent we have studied, because we need to validate it, right? Whether PAD4 inhibitors can be antifibrotic or not. We have proven this, and we have published this work in the International Journal of Pharmacology. Now we know that netosis is important, and we also have an in vitro model where HL60 cells, which are the leukemic cells, we have stimulated with the PMA, because they need to be leukemic cells need to be differentiated to neutrophil like cells. That's why we have used the PMA, pore ball minister 30 states. The green color is nothing but more nets are formed compared to the normal control. When we stimulate with the PMA, because we need to have a proper model, right? We have stimulated PMA, we can see increase green fluorescence is nothing but netosis markers. This is actually the video, time lapse video, where different groups, control, PMA one, and also a treatment with our compounds. we have seen, no, this is the green. Actually, these are the videos. The video was not playing. I had tried. So what we seen was, no, it's not just one image, right? If you can take a video how the nets are formed from the normal cells to the uh, the cells which are undergoing netosis, we have taken this image, so clearly evidencing it, and we have done the since we have this particular the AIDS model as well the polymorphism model. We also do the nanoparticles. I mentioned about the smart nanoparticles. We have demonstrated that smart nanoparticles are controlling the uh, ARDS, which is acute respiratory distress syndrome. These are some of the lung functional parameters as the lung images, where because we are coating with the DNAs, they are controlling it. This is histological data indicating that nanoparticles are smart nanoparticles are controlling the severe lung inflammation as 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 equivalent or better than the dexamethasone, which is a standard drug. Now, after confirming that no, there is a way you can control the netosis. Now we try to work on the novel PAD4 inhibitors because chloramine, which is a experimental compound, cannot become a drug. That's why we have is the no, we try to discover the new PAD4 inhibitors, and for that we have to in silico and cell culture studies. So initially we have tried to understand the protein structure. What are the PAD4 structures available in the literature? Because you need to take in the initial slides I mentioned, you need to take the right protein structure. If you don't take the right protein structure, what are the work you do maybe not proper. That's why you need to analyze the protein also. There also you can use the protein, but no, that may be giving different types of protein. We need to pick the right protein. You need to validate the protein structure which you are taking is right. We have done this particular study, and we try to use all the 4 different protein structures available in the literature with the different PAD4 inhibitors reported by different groups. If you see, you have a GSK, it's brittle Myers Squibb, and other companies are already working. Why? There are Jubilant Bio also working. So many companies are already working and they already have some molecules discovered. Why they have discovered? Why they are investing a lot of efforts in this? Because they realize that PAD4 can be a ideal good target for to control some other inflammatory condition, even the cancer also. Now, how this PAD4 known PAD4 inhibitors developed by the different companies are having a different hydrogen bonds and pi stacking interactions and all we have analyzed, because you need to understand the existing data how they're binding. If you don't understand the existing interactions or known inhibitors, you will not able to give the proper command, right? Okay, I want to have this many hydrogen bonds, I want to have this particular property. So you need to because of this particular bond, this the compound is behaving like this. Now if I can increase the bond or decrease the bond, what kind of interaction my new compound is going to have to the target can be understood. So again, this is the docking of the known chloramidine, which is the known PAD4 inhibitor, as well as GSK molecule to the protein structure what we have optimized, and again chloramidine with the other protein structure we understood. Later we have designed the molecules in collaboration with the chemistry. Then followed by we have screened the molecules PAD4 inhibitions. We have a Docker score based on the computer generation. We have the PAD4 inhibitor assay kit. We have done this particular study. We also understand how they're killing the cells. And we also understand what is the amount of the netosis which is inhibited. We have identified that one compound called 36E at the molecule 14 has shown better activity, better inhibitory activity on the PAD4 compared to the existing chloramidine. So we also further understood how it is exactly doing the molecular simulation dynamics by running the simulation studies by using a Schrodinger software, and we found that it's having the required hydrogen bonds and other the bonds to bind to the PAD4 to inhibit it. Then followed by okay, sorry, took some time. Wait. Kind of stuck actually. I'm just okay. So and again we understood how it's binding to your the PAD4 inhibitor for the docking studies. Then we did the in vitro cell culture based studies, how the react toxin species are actually no, not react toxin species, how the nets are actually in nets which are increasing the PMA, and our compounds have decreased these nets. And this is the microscopic images of the cytox stain, which is used to understand the net's the status. Very bright green fluorescence coming from the positive and in the sense PMA treated disease cells, and treatment with our compounds have controlled this intensity. That means our compounds are working. We also done certain the DCFD and myeloperoxidase and other assays which are related relevant to our PAD4 inhibition. We also done the immunoflow cytometry for cetylated H3 histone protein, right? Cetylated H3 histone 3, which is increased in the cetylation is the process where your netosis is taking place, right? We can see the in the PMA state itself the more red fluorescence is that nothing but cetylated proteins which are reduced upon treatment with our novel inhibitors. The followed by we did this our novel molecule in the AIDS model where LPS will be injected into the lungs, which is causing a lung damage. So these are the functional parameters, lung images showing that no lung damage has taken place because of LPS, lipopolysaccharide. Our PAD4 inhibitors protected it. These are histological images demonstrating that LPS, which is causing a severe inflammation, as well as this blue color staining is nothing but the toluidine blue staining, which is used for the mast cell infiltration into the lung, which is also demonstrating that there is a severe lung inflammation due to LPS. Because you need to have a proper animal model, then only you can able to study your compounds given through IP route as well as oral route as control. The what compound I'm talking about my PAD4 novel inhibitors. We have demonstrated it. Then followed by we also demonstrated its activity in the pulmonary fibrosis model, where again we have seen a product activity through the different pro-inflammatory cytokines and pro-fibrotic cytokines. We also done the histopathological studies, HN staining as well as mesenteric staining, which is specific to collagen. So in disease control, we have severe extracellular matrix accumulation. So the alveolar structures which are required in the normal control are all blocked, are all narrowed down, so destructed. And chloramidine and our PAD4 inhibitors, 3 mg, 10 mg, and 30 mg oral route have actually protected from the fibrotic condition, and they are much better than the standard drug like perfenadone as well as the chloramidine. So we also done the immunohistochemistry study to understand the collagen expression, fibronectin expression, and the PAD4 expression, because these are the markers we hypothesis that have a role in the pulmonary fibrosis. And followed by we also done the another inflammatory model, which is a acute pancreatitis. Acute pancreatitis generally seen in the alcoholic people. Acute pancreatitis, you can go back and read about its severity. It has high mortality. Basically, it produces systemic systemic inflammatory syndrome, systemic inflammatory response syndrome, SIRS. So when there is acute pancreatitis, person is 50% in the danger of getting getting losing their life. So that much severe is the acute pancreatitis condition. We have a model to we have an animal model, cellular lineage animal model. So we have developed this model and try to understand, because again inflammation is mediated through the netosis. If whatever the PAD4 inhibitors we have, we try to study, and we have seen that no, they are protecting the acute pancreatic condition. So different biochemical as lipase and amylase and lipase and histopathology and so on, we have done it to prove that no, actually they have a protective activity in the model. So another target we have taken was a certain 3, which is a NAD dependent protein deacetylase, which is required for your mitochondrial homeostasis. That means it's actually SAT3 is required for your endogenous antioxidants like a superoxide dismutase to properly function. The certain levels are decreased. That's where you see severe oxidative stress will be causing a mitochondrial dysfunction, and which is responsible for the cell death in acute models, acute conditions as well as the chronic conditions. We try to study how this certain SAT3 activators. Now we need to have SAT3 activators will have a role. Now if this is SAT3 activators, we have resveratrol, honocoil, and these are natural products. We try to study how the synthetic SAT3 activators are effective in fibrosis. So C12 is an existing molecule. C12 is a chemical name, a coumarin derivative. Already known SAT3 activators. We demonstrated how they're actually protecting from the lung damage in vitro, and also how the cell death in the lung epithelial cells, BS2B cells, are we see that no C12 protecting the your lung epithelial cells from the damage induced intentionally. And in the animal study also we have proven that the SAT3 activator C12 protected against pulmonary fibrosis. Again, this another study. Again, we have published this work in the paper. Now since it's SAT3 is a mitochondrial protein, we try to target the this particular C12 to mitochondria. That's called mito C12. How we can do is we can actually have the TPP, tripenyl phosphorium is the chemical which is having a highly cationic, because highly cationic molecules can go to the mitochondria and accumulate there. Now if we conjugate C12 with the TPP, the TPP will be driving your C12 to go to mitochondria, so that you can reduce the dose and it's going to be more effective compared to the C12 alone. This is the strategy people have been trying it. Now we can only use this strategy to take the compound to the mitochondria. It's basically mitochondrial targeting approach. And because of mitochondrial targeting, mito C2 is doing a better activity compared to the C12, which is a known C12 activator. So based on this success, now we try to develop the new SAT3 activators, again in collaboration with the chemistry. These are the docking scores. They're not the dock, these are the in the screening, where we have either SAT3 assay kit. Many compounds are active at the 5 to 6 micromolar concentration, which is good. And this is how they're having interaction with the SAT3 protein. And another series done by other faculty, because we cannot just depending on one series. You need to have multiple series to have a better molecule. The another series we are developing, and all the molecules are in the range of 3 to 5 micromolar concentration to activate SAT3. And whereas the C12, which is a known SAT3 activity, is much less compared to our compound. So we have actually we have done the animal studies as well with these compounds. Now actually we are going to patent this. Unfortunately, these molecules, both PAD4 inhibitors as the SAT3 activators, we have not used the AML tools. We just use the Schrodinger molecular modeling. We have not optimized for the AML. Had it been we have used the PAD4, the AML tools, we would really might have got a much better lead. These are the initial the results. What they're telling is, yes, we have a new compounds designed, synthesized, and evaluated. However, we need to further improve. How are we going to? We are going to now optimize the chemical structures by using our AML tools. We are now collaborating with the experts in this particular field from the industry. They're going to design a molecule for us, so that we have the tools to screen in a biology models, and we have the tools to synthesize here. What is required is a designing it. Now, if one of us or some of us having this expertise, we can able to play with this softwares, able to understand these basics of the AML, will be the hard cake to collaborate with so many people. And also, trust me, there is a lot of money for such kind of people to start their own company or do the consultancy services. Right? I really wanted some of you to take up this as an opportunity to learn the AML tools, drug discovery, and I think will be highly successful. So another target we also have taken neuropilin 1. It's a biology I don't want to talk about that much. And neuropilin also having a role in COVID virus entry. And we also know that now because of COVID, long-term complication of the COVID is lung fibrosis. And incidentally, we have been working with the COVID as we have been working with acute respiratory distress syndrome, which is immediate effect of the COVID, and the chronic effect of the COVID is fibrosis. We have been working with both ARDS, which is acute condition, pulmonary fibrosis is the chronic condition. If you see the science paper which came in 2020, it's clearly mentioning that neuropilin receptors are co-receptors for the virus to enter. That means along with your ACE2, even this is also a co-receptor for the host cell to accept the virus. That means if we can develop the neuropilin inhibitors, it is going to prevent the viral entry, and it is also having a role in fibrosis. Because when we did the animal studies, when we analyzed the normal control and the fibrotic samples, see the neuropilin expression. Neuropilin expression is highly increasing. What it indicates, that means, okay, this particular protein, which is required for the viral entry, which is we have seen the science paper, is also contributing to the fibrosis development, which is we have done in the our mice models. So what we believe is, I think we can design the molecule. We can, if we don't have a proper neuropilin inhibitors, if we can develop the neuropilin inhibitors, though we are not planning to the viral inhibition studies, that can be done in the collaboration. But our models, our tools are here fibrosis. We want to understand. One of my PhD students is trying to work on this. Now we are trying to develop the new neuropilin inhibitors, so that now we'll be able to study how they're protecting the pulmonary fibrosis. That's what we are trying to do it. So I think, if you see it, as a pharmacologist, I cannot do alone things. You need to have interdisciplinary teams. You need to have the teams from outside your specialization, where you can collaborate, you can partner, whatever the way you can, get outsourced. If you have the funds, you can get outsourced and get the work done. Just see, a company which is in Taiwan, company which is in Korea, they are trying to come to Hyderabad. They never visit Hyderabad. They just pay some money and say that, no, look, I have this particular target. Can you design the molecules for me by using AML tools? Yes, I will pay 1 crore rupees. Okay, we are working, since we are getting 1 crore. You will design the molecules and give to them. Now, the same company will go to another biological and pharma company, say that, no, look, I have this 200 molecules. Can you screen these molecules for this particular activity and give the results? Yes, since I am getting, we are CROs, right, in Hyderabad. Okay, I will do it. Now you'll give the results to them. You are not, because they don't disclose. It's in different segments, right? Designing is done somewhere else, and screening is done somewhere else. Now, based on the results, what that person will do is, they try to go for the IP, patent. Once patent is done, now whatever investment you might have done is going to be multifold, right? Because it's a new target, you have a new molecule design. Because you have used AML tools, you could able to get the better molecules than the existing ones. And you also have a pharmacology and biology data and target based assays saying that, no, yes, it is actually having a activity. Now you do some safety studies in the GLP, if you have funds, and go for patent, and anybody is ready to take your molecule. If they don't mind paying few million dollars, at least, it can be few million dollars, $10 to $100 million, it can be. There are people who have been enjoying this. That's why, we need to have a teams done together. For that, what is required is, you need to adapt the new tools. You need to read more. We need to understand more pharmacology. We need to understand more disease pathology. You need to read the new novel targets which are emerging, and what are the problem with the existing drugs. If we can spend a lot of time on that, I'm sure we will be able to come up with something novel. So what I want to suggest is, okay, so you may be having a molecule, you may be having some lead compounds, but safety is very important. So you can also have an idea, and all the safety studies, all the toxic studies can be optimized by using AML tools. So toxic studies are required. You are going to optimize all the toxic effects, because you have taken care of all the structural hints which are going to be causing a genotoxicity, hepatotoxicity. This all you can optimize it with AML tools, so that, no, we'll be able to do it. Now, at the end, what I want to say is, all the faculty, don't just think that, no, we are only going to teach. You need to adapt to the new tools, at least the basics of it. They are really going to change your job opportunities. I don't want you to be always in the same job. If you are enjoying it, well and good. If you are doing research, well and good. But yes, you should have your own way of growing it. So AML can be one of the potential medium for you to go now. Now, if you want to work in your laboratories, different things. Me being a pharmacologist, I'm proposing some of the topics. You take a model, so because chemistry good, you can able to have a chemistry target, you can able to take it. The one of the biggest problem for many of the farm faculties, they will not have a good model to study it. So take a model which is simple to develop it, like psoriasis, allergic contact dermatitis, burns, wounds, arthritis, inflammation, lung disease like ARDS and lung fibrosis, renal fibrosis like unilateral ureter obstruction, which is a surgical model, renal damage model like cyclosporin, cisplatin, and amphotericin B induced models, hepatic damage, paracetamol, C-cell model, and many more. Like that, you take a model which is very easy to develop. You don't need very sophisticated to do it, and the chemist can give the molecules to you. The formulation people who have developed a novel formulation can give the molecules to you, so that this all can grow together. A publication can be originated, a patent can be originated, so our new molecules can also be designed. That's what is possible with the departmental collaboration within the institute as well as the outside institute. I think with this, I would like to thank the you all for kind attention. I know it's a one-sided, it's a monotonous. I hope I have conveyed something to you. I would like to take few questions if you have, and like to thank again for your kind attention, as well as thanks for the opportunity. Thank you.
Thank you, sir. Thank you for
Over to
this.
you. Over
Thank
to
you,
you.
sir.
I hope I not bored you.
Yes, sir.
I'm
No,
also
sir.
sorry.
Not at all. Not at all. We learned a lot, sir, from your vast experience and the extensive research. What you have done is really meaningful for the society, because as you have came across from some AI tools you are using to identify the target, and now then you have decided to work on the animal models, and definitely you have get a very good result. We can see the results and how it can be useful definitely for developing a new drugs. So in your talk, you have discussed about the basic of drug discovery and drug development, and also how you can AI ML can be a startup. Also, you have discussed in your discussion of your presentation, and also you have nicely discussed about the Nimolide, how exactly you have taken it and target the TNF alpha. It's really commendable, sir. So I have a few questions from my side
Yes.
first, and I'll take some questions from the audience.
Yeah.
Which are
Sure.
the which are the softwares that you used, like docking software for this Nimolide, to identify the target, like docking software?
We have used the Schrodinger, which is actually
Schrodinger.
we can also have the cracked version, but our
Okay.
institute we have this the official version.
Yes,
Yeah,
yes, yes,
Schrodinger is the one. Yeah.
yes, yes. Okay. And
Yeah,
sir,
I
is
can
there
see a
any?
lot of people, saying thank you, the
Uh,
appreciating the session. I would like to thank all of them. Thank you so much. Yeah, yeah. Yeah,
yeah. So,
carry on with the questions.
sir, is there any other tools which is the freely available tools for this identifying the target molecule as a docking software? I'm asking
I think
Autodeck, Mina, and all.
I think Swiss tools are there. Swiss suit is there, as well as we
Okay.
also have Autodac Vena. These are some of the tools,
Yes.
because
Yes.
since we have Schrodinger, I have not invested a lot of energy to understand what are the free tools, but they are available and people are already
Yes.
using and publishing it actually.
Yes. Yes. Yes.
What I want to suggest is, just don't do peripherally, go more deeper, understand more deeper. There is a lot you can optimize it. You need to spend a lot of
Mhm.
time. Let it be a stupid activity, but do a lot of in-depth, spend a lot of hours. That's where you'll find some of the interesting things. Small clues only give you big breakthroughs, I feel.
Yes. 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 And here you have rightly said that collaboration is very needed this time,
Highly
because there's a
essential. Highly essential.
yes, because in pharmacy we are not have that much of techno friendly, so we can have we some friends from computer science who knows the AI, so who knows the algorithm, so you can give
Not for the
them
sake,
that.
not for the sake of certificate. You need
Yes.
to
Yes. Yes.
do the practical aspects of it.
Exactly.
If they
Exactly.
can and really handle those, I think a lot of demand. Trust me, a lot of demand. For the
Mhm.
job opportunities can also increase, because all the pharma companies will be now going towards these tools. It's a very
Mhm.
good activity to learn, basically, because we are
Okay.
very good in our subject, but no, that may not be sufficient now.
Mhm. That's true. So, sir, can you just open the chat box? You can also get some questions over there. So, you can take some questions from them
Yeah.
in the chat box. You can open from your side.
See, I already mentioned the free tools for the softwares for molecular dynamic simulation, as well as PDPK modeling. So you can have Schrodinger, the cracked version, as well as the Autodac Vena can be one. You can also Swiss tools, and I'll share you. Otherwise, and I think I need to get this information. Some of the free tools I'm not sure. Schrodinger is the one we have been using it, but there are a few more also. I'm sorry in case if I'm not able to give the full list of it. please enlist the AML tools that you may have used in your discovery pharmacology research. Actually, we have not used it. We have collaborated it. Because no, again, this is highly upcoming area. We have collaborated it, but I wanted you people to learn it. I wanted you people to learn it because I'm asking you to start ABCDs. I'm asking to start ABCDs. Okay, we are not late. If you even start today, down the line after 1 year, you'll be having expertise. And actually, I was paying 5 lakhs rupees just for one design, but it's really crucial for me. I don't want to waste a lot of my time just doing the pharmacology, doing chemistry with unnecessary molecules. I want a molecule which are very intelligently designed by using ML tools. There are companies to do this, but they charge you, right? Or you need to collaborate with. Yeah. And I don't. We have more questions.
You can just scroll up, sir. Scroll up. There are some more questions is there.
People have already left. Looks like.
Okay. No, 193. Look, people are there still. I can see there are 193 people are there. 193.
It's a big number. I should take a. You should be very happy for such a wonderful response. I'm glad that some of the people have liked it. Thank you all for wonderful feedback. But I'm not giving a talk just for the feedback. If that has helped you, I'm more than happy because my job is only to convey that there are opportunities. I don't want to claim that I'm great, but we are also learning, and few of the things I also don't know. I may have limitations, but you can actually invest your energy into that, right? That's what my intention is, right?
So if anyone like to ask some questions, you can unmute and you can please ask some questions.
Yes.
From the audience, you can unmute if you have any question from your side.
Great. Yeah. Mehta, Danmay Mehta. Okay.
Oh. Please, someone, if anybody is interested to ask some questions, you can please ask by unmute. Dr. Madhavi, madam, you like to ask something?
Yes, sir. Am I audible?
Yes, madam. Yes, madam.
Sir, I'm from the Department of Pharmaceutics. Okay, so some things I was able to observe. But my query is on, you are telling that AI tools have been generated at various, even for the screening of the target, right?
Yes.
So my doubt is, you were talking about data sets that have been contributed from, all the researchers, so far that has been happened over the years. So how is this, who is doing the training for these data sets so that we get the right, selection? Because just if you see, sir.
What I say, nobody will give you training. You need to use the informatics. You need to know. So all the networks analysis you have to do through the informatics tools, pharmacoinformatics tools. You will not able to do. You will not able to learn. But that's where you need to collaborate. You need to collaborate with them, right? Yeah. Because you only reading the articles may not be, even if you are reading the article also, if we could able to analyze through the informatics tools, confirming that okay, what target I'm taking is making sense.
Now, why? Because when we use this for research writing and all, when we are using the paraphrasing tools or something, yesterday's session also Dr. Akash was talking. So unless there's a subject expert, we cannot identify what are the mistakes in the data which AI is giving us.
You need to
so like that.
Yeah.
So my doubt is when we are trying to use AI for the, let us say, getting a lead molecule or something from the 20,000 you were mentioning, how much reliable is that tool that we are using?
But we are doing the biology, right? It's just, you are avoiding what are the bulk to be done. And see, I have 100 molecules. Maybe 90 are not able to meet my requirement. At least 10 are meeting my requirement. I have filtered out from 20,000. I can get 100 molecules which are actually meeting my criteria. My criteria is to have these bonds, these properties, Lipinski rules. So the rules set by us.
That information we are feeding to the system.
We have to give the input. Yeah. we are putting the filters to get our desired molecule, right? You can also do the SAR, structural active relationship, without even doing the study.
Yes.
So these all we can use the tool.
So how would this AIML be different from the current chemistry? Because we used to have chemistry tools, right? Maybe 5 years ago, 5 or 10 years ago.
Uh-huh.
How are they different from this AIML? I
Because
can't name it because
if you only just use the docking tools, your they are also kind of AML tools only. But no, the
okay.
advanced tools like bulk data, if we can analyze through the
So
neural
the
network and
quantum
other
of
programs,
the
that's where
quantum of data we are processing.
that's what happens. One more advancement in the molecular modeling is the AML tools. So you are using a Schrodinger or any other tools, you get some information. Now, based on that, you are trying to further optimize by using AML tools.
Okay. So the quantum of work is being reduced for the scientist.
Exactly. So but accuracy also increased, right? Accuracy will be increased because you have, see,
more data is at our disposal.
The moiety will be done 3-dimensional. All the its alignment, its interactions, binding, stability, all can be studied here itself. But it was not the case. Earlier, we need to do the experiment to prove that, right?
Yes.
And
yes.
we are investing a lot of energy on the molecule which you are not even sure whether it's going to bind to your target or not.
But it's going to come or not.
Yes.
Now we are able to predict all that with this.
At the computer level, we know that this is going to talk to the target, bind to the target, and having a desired properties or not. If not, what is required, I will try to modify it. I'll try to modify my structure. And again, I see what is my docking score. If because of modification, is still binding or not. Is because of this, any toxicology features increased or decreased. So you optimize it.
Can you suggest
Yeah.
Please continue, sir.
So it's like multiple parameters can be optimized. Which is we cannot do it with our own manually, right? That's why a computer is going to be. Yeah.
So I mean, with these tools available, because chemistry wise, how quick can you get a paper with AIML?
Think beyond paper, madam. Think beyond paper.
No, my understanding, because we are handling PG students, right?
Yes.
For them, it's like, fingertips stuck to every, maybe 1 week or something, they're coming out with a paper. So what is the actual quality of that work, and is it really possible to get this is my query.
But discovery is publication. I have my own limitation. It can only give you the information, but you need to write only. Publication, I don't think so. You can actually use the tools, and even the publishers are clearly mentioning are using AML tools to write this paper. That's why it's only
not for writing, for the discovery part, for the discovery
yes.
part, or a model, any structures that way.
Okay, what is the question?
No, using this AML tools, if you want to get a paper on a new molecule, how much time does it take?
Okay, madam, if you have a good tool, without even synthesizing, if you do the molecular simulation MSD studies, we can able to publish paper within. You can complete the work within 1 month. I have seen people completing the study in 1 month.
That's what.
You know to do an experiment. You only have to filter the data and from the different data sets and analyze them and making a meaningful conclusions made.
Yeah, so you just need to work on this AI and softwares, and you'll be able to get a credible one. It's not, I don't say substandard something, but of good quality, definitely.
No, good quality. No, actually, my student is a pharmacologist. He's in the 3rd year. He had to start his annual pharmacology. He had to do pharmacology studies. What he's doing is trying to understand these tools. Just imagine, pharmacology person investing a lot of money just to optimize.
Understand, yes.
And he wanted to publish a paper. I said, no, don't publish papers. Wait, we need to
Let us study first.
We need to get it edited by get it read by expert from that side also. Then only we can go for it, because you may be doing a unknowing mistakes. We don't want to do the silly mistakes, right?
Correct.
So though he is doing a lot of studies, we still collaborate with outsiders.
Validation of our work.
Yes,
from the external agents.
Yeah, you use the right word, validation. Yes.
Okay, sir. Thank you so much.
Yeah. So you're from the same institute? Which institute?
I'm from Dhanvantri, sir. I'm from Bangalore. Actually, native of Vizag.
Okay.
Presently in Bangalore.
Okay. Nice meeting you, madam. Thank you. Nice talking.
Thank you, sir.
Thank you.
Okay. Thank you, ma'am, for asking a question. Now I can say Dr. Ashwini Mishra. Please, you can ask your question.
Hello, good afternoon, sir.
Good afternoon, Ji.
So thank you very much for such a nice presentation.
Thank you, Ji.
I wanted to know that whenever we are collecting the PDB ID or PDB format of the protein from RCSB, what are the validation protocol we have to apply in order to validate the structure of the protein which is deposited in PDB?
Okay. So actually, this part I have not done it. This part I have not done it. I can get back to you. To be frank, I can get back to you on this because so what data we got is my students have done it. I'm just presenting it because I'm a pharmacologist. I have not done this, but my team has done it. If you want me to connect, I can connect so that no, you can take my number. You can have a talk.
Okay.
Yeah.
There is one more
I'm
question.
sorry in
Like,
case because I cannot able to
no,
answer.
it's okay, sir. I can
Yeah.
understand. Nowadays, like research is a multidisciplinary work, and
Yes.
where there is involvement of the many people in order to generate the perfect output of
Exactly.
the research. So one more thing, it was there, like probably before a couple of days, I was into evaluating certain kind of research work where a question came to me that whenever we are using any kind of ligand in the case of molecular docking, and suppose like the take example of diclofenac and diclofenac sodium. So whenever we are applying the pipeline of the molecular docking, we normally go for removing the ions from the molecule and then we docked it with the protein. But when it comes to the conversion of the nucleus by enhancing its solubility by the application of ions into it, is it that whenever a molecule binds with the receptor, the ion or the partial charge which we have associated during synthesis will also attach with the molecule or not?
Excellent. Okay. So I think we don't use ion channels, and I think it's very good question to have. Yes, what your question is, ion channel also having a the role to interact with the protein or not? Don't no need to check. I think we it's only physical chemical properties we're trying to alter it. I think it won't participate in the interactions. It's all the core moiety, which is not the salt, which is going to be having role. I think, but it still needs to be evaluated. I still need to be checked. I think somebody can do this. Um, I'm not sure on this.
Yeah, because if it doesn't work or probably the docking process do not involve the ions along with the main nucleus, then docking is not at all suitable chemical investigation field for those molecules which are actually available into salt form, because we are removing the salt before forming the cluster between the ligand and the protein.
No, see, salt form mostly for solubility issues, right? Salt form comes with solubility issues, but when you do the discovery, you're not going to see. You don't use aqueous as a your solvent for you to do the in vitro studies. You're okay even still to do the DMS as your solvent to do the study in vitro studies. I'm talking about. So yes, if salt form is not the first priority, when you try to make a salt form is when you have a solubility is actually maybe troubling you later that you are trying to design now itself. Now, I think that can be also docked with if you are doing the salt form, that can also dock with your target. How what kind of is the interaction compromised or not? The interaction is not compromised, is okay, right? Interaction should not be compromised. The target and the ligand interaction should not be compromised because of the salt. If you can prove that is fine.
Okay. No, that is the thing ultimately, sir, because if it is not going to be compromised by using the salt form, but during, like as you before a couple of minutes you said that people are using autodock vena. In the case of molecular docking, where first two step in the preparation of the ligand molecule is to make it charge free, and the other one is we are actually removing the charges, even the hydrogen in a way. So if we
Okay.
are removing everything from the ligand, then how come the salt can be utilized in the process of the docking in order to understand its energy of the cluster formation?
Okay. Mm-hmm.
That is the point, because I've been thinking a lot for this particular problem, and whatever protocol I've been using, the thing comes is that you cannot take the ion along with the ligand in the case of docking. though in vivo condition it might be possible. In the case of these kind of machine learning tool, it is really a big doubt that whether the ion is going along with the
अरे
ligand
मैडम वो
molecule.
बच्चे को कहाँ
I,
बिठाना है
I,
मुझे
okay.
इधर
I think you need to talk to the experts in on this particular on this particular I think I understand you are working in this and you're facing it. I think I mentioned as a pharmacologist I don't have this much deeper. What I'm only supporting is we need to go together, but I think you need to talk to experts. Maybe that they can give you a hint on this. I
Thank you, sir, for
Thank you
asking
very
this
much, sir.
question. Thank you, sir.
Yeah, no, actually, yeah. In fact, though I was, presenting the molecular modeling and all the simulation, this this is the part done by my collaborator and my student. So I don't have a lot of, the background. So that's where, I could not really answer it.
Okay, thank you, sir. So we can take one or two questions. So anybody
Yes,
want to
yes,
ask a question, you
yeah.
can please ask. Any audience?
काम कहाँ है
Dr. Abhay Pratap, you would like to ask some question? No. Okay. Any more question? Chandra sir, can you please stop sharing from your side that slide?
I can do that. Yeah.
Okay, okay. So I think there are no more questions. So thank you, Chandraiah sir, for such a profound and engaging session. Your insights into drug discovery and development, the era of AI/ML, is really meaningful. So now we are proceed to the certification process. So kindly just give me 2 seconds.
Did I close this sharing?
Yes, sir. Please, you can close.
Okay.
Just 1 second.
Do I do something from my side?
No, sir. Just stay,
Okay.
sir. 1 second.
Okay. Okay. So very good attention, man. It's 177, 190, more than 200.
Yes,
No, that tells the spirit of the faculty. Now,
sir.
with this, with this course, I think they should charge up and learn this challenging task.
Yes, sir. So, sir,
Thank
this
you.
is on behalf of PP Savani University. So, please accept this certificate of appreciation as a token of our gratitude and recognition of your invaluable contribution.
Thank
Thank
you
you
so
again,
much.
sir.
Thank you.
Thanks a lot, sir, for your insightful session. And I
Thank
will,
you.
most of us
Thank
is
you all. I wish you all the best. Wish you the rest of the program all the best. And also, I really love to see people, grab this, this challenging area of AML and, become experts. We are not late and we are not lagging. I think you can start now at yourself.
yes, sir. As
Thank
you
you. Thank you for the certificate as well. Thank you.
yes, thank you, sir. Thank you, sir.
Thank you.
So now I like to all the participants. Kindly, I'm going to share the attendance of today's and feedback. Kindly fill the feedback form in the chat box. Just give me a second. I'm going to share the attendance.
So I'm done.
Yes, sir. Thank you, sir. Thank you again from our.
Thank you. Thank you. Thank you, Bhaveji. Thank you.
Thank you, sir. Thanks a lot. So all of you can you just turn your camera on? You can take a snapshot.
What is required?
So I am requesting all the audience to turn on their camera, so I can take some pictures.
Okay. So I'll be there also, right?
Yes, yes. Please
Yeah.
stay here,
Yeah,
sir.
I'm there. Yeah.
So I'm going to take some pictures. Just be here.
Thanks, Shilpa. So you see, gave a just good session. So thank you.
Yes,
I
sir.
could see the message.
So 1 more second. I'm going to click some photos. Okay, thank you. Thank you everyone for your kind attention. So tomorrow we are having a session from Dr. Javed Ali, the famous person from the Jamia Hamdard University. So he's going to talk on about our publication regarding session. So all of you please kindly report by 9:45 a.m. tomorrow to learn something new from our beloved Dr. Javed Ali sir. He's a renowned professor and head of Department of Pharmaceutics, Jamia Hamdard University, New Delhi. So thank you everyone to joining our session. The feedback and attendance link is there in the chat box. Kindly fill it and then you can leave the session. Thank you. Thank you everyone.
Thank you.
Thank you. Thank you, Chandraiah sir.
Thank you.
Sir.
Thank you. All the best. Thank you. Good. Well organized. Well organized. Good.
Okay, sir. Thank you, sir.
Good. Good. Thank you. Bye.
Thank you, sir.